The overall goal of this procedure is to label single neurons in the central nervous system of early stage 17 Drosophila embryos. This is accomplished by collecting eggs and allowing them to develop to the right stage. The second step of the procedure is to manually decorionate and align the eggs. Following this, devitellinization and filleting of the embryos makes the nerve cord accessible. The final step is to label single cells by iontophoretic application of the lipophilic dye, dye-I. Ultimately, results allow the visualization of single neuron morphology, if desired in the context of known GFP expression patterns and/or in mutant background.
The main advantage of this technique over existing single-cell labeling methods like Markham is that it works in the embryo and allows to visualize identified cells intentionally.
Visual demonstration of this method is extremely helpful, as many steps are delicate and therefore difficult to learn.
Allow healthy flies to lay embryos on agar plates. Replace the plate every 1.5 hours at 25 degrees Celsius and retain the embryos. Incubate the embryos on their plates at 25 or 18 degrees Celsius until they reach the required developmental stage. When ready, transfer the embryos to a slide covered with double-sided sticky tape. Avoid transferring any agar with the embryos, as it will interfere with their adhesion to the tape. On the tape, allow the embryos to dry for 5 to 10 minutes. While waiting, coat a 24 mm by 60 mm